All right. Okay, thank you all for coming. So let's start. It's my pleasure to introduce Adrian Jacobo today. Well, the oldest ones, probably you know him, the youngest not, maybe. Adrian is, well, he is originally from Mar del Plata, but he did the PhD here and the co-supervision of Pera and myself. He was actually the first person I co-supervised. So I'm glad I made no harm, and he progressed on his scientific career greatly. So I'm very proud of him. Welcome. Well, in the PhD thesis was about pattern formation in nonlinear optics. But then, after when he moved as a postdoc first to the Max Planck Institute for the Physics of Complex Systems in Dresden, he shifted focus to systems biology or developmental biology, or the effects of pattern formation and nonlinear dynamics in the developmental biology. He spent there 2 years as a postdoc, or almost 2 years, and then he moved to Rockefeller University to work in the group of Jim Haspeth, which incidentally I just knew that he passed away last summer. So I'm very sorry for that. Yeah, I knew because Adrian told me. Adrian spent there a few years and as a postdoc, and he developed the skills and the knowledge he knows about environmental environment and embryo development in biology. And then in 2022 or 2022, he got a position as a group leader in the Chan Zuckerberg BioHub in California, in San Francisco. So, well, we are very, very happy to have you here, and he will talk today about how do cells count, cell number control in regenerating organ.
Thank you, Tamia, for the kind introduction. And yeah, I guess it's nice to see old and young people, people who I haven't who weren't here during my years at the FISC. And yeah, I guess as Damia introduced, I mean, I'm still interested in pattern formation, and I think sort of like developmental systems are sort of a prime example of complexity and pattern formation. Is something that I'm very interested in, is what I'm pursuing in my group. And so just maybe for those of you who don't know, what's the BioHub? So the BioHub is now starting its second 10-year tenure. So the first 10 years were like, basically we got a bunch of money and we were doing whatever we wanted. And for the second 10 years, they asked us to say like, okay, what is it actually that you want to do? And so our mission is to illuminate and predict and program biological processes in real time and across scale. So predicting and understanding biological systems has a lot to do with like modeling and understanding a complex system and a system that in particular has a lot of spatial temporal structure. So understanding how that structure arises sort of is fits square within our mission. So this here is our current building. We're moving in a year and a half to a new building in Redwood City, but this is in San Francisco. So it's the skyline of San Francisco here. And so this is the Chase Center where the Golden State Warriors play. Yeah, this is where we are. And then like in a building that is around here is where our fish are located. As Damien mentioned, I did my postdoc in the lab of Jim Haspeth, where they study mechanosensory epithelia, in particular the workings of the inner ear. And when I joined the lab, I thought I was going to do something completely different. I thought I was going to work on sort of like the biomechanics of how the hair cells, which are the mechanotransductors of the inner ear, work. But then I started seeing some of these images. This is an image of a rat cochlea, and some of these where you see sort of like the organization, the multi-scale organization of the mechanosensory epithelia. So this is the different rows of mechanosensory cells that are surrounded by these pink cells, which are support cells, and you see that they form this very regular hexagonal arrangement, and there's like 10,000 rows of these along the turn of the cochlea. Each one of these cells is different. They all have these structure, which is called a hair bundle, which is actually a bunch of cilia. And these cilia are the ones that allow you to decode the sound of my voice and convert it into electrical impulses. And each one of these is slightly different along the turn of the cochlea, and they're tuned to different frequencies and so on. But this, so all of this is to say that this is a system that is able to form these exquisite patterns that are coordinated across multiple scales. And so when I saw these images, I was like, oh, this is what I want to do, actually. I don't care about the biomechanics. So, and Jim sort of like, he was like, yeah, do whatever you want, basically. But studying the cochlea is very hard, and in particular the development, because it's an organ that develops encased in bone. So you can't see dynamics. So you can only see snapshots. So it's very hard to reconstruct dynamics from that. And I also don't like working with mammals. So so we work with zebrafish. So zebrafish are these small fish which are usually used in they're very common in the pet trade and have become sort of a model organism for vertebrate development, in particular because when they develop, they're transparent. So you can put them under a microscope and you can take these sort of beautiful images. This is from my colleague Loic Royer. And so this is an embryo which has all the nuclei or the cells are fluorescently labeled, and you can see sort of how the embryo develops and forms structure. So you'll see the head forming here and the tail, and the somites, which are going to be sort of like the structures that then give rise to vertebrate, the vertebra and the ribs. And in particular, so zebrafish have an organ called the lateral line. So this is a series of sort of patches of cells called neuromasts that are right underneath the skin, and they're an evolutionary precursor of the inner ear. But instead of using these to detect pressure waves inside the cochlea, they use them to detect the water flow around the fish. So the cells here are sticking out in the surrounding water, and they can detect, they use them for proprioception, so they can measure the velocity compared to the water, but they can also sense if predators try to eat the fish. Usually they try to suck them from behind, so they measure this water flow and they can escape. And they also use them they're very sensitive, so if there's prey, so this small paramecia the fish eat, swimming, they can calculate sort of like the velocity from the vortices and turn around and eat them. And what happens is that so this organ develops underneath the skin and the fish are transparent, so we can put them under a microscope and take some of these movies that recapitulate most of the developmental trajectory of the organ. And that allows us to use computational tools, so we can segment and track all the cells, and we can create sort of like a virtual representation of the organ. That then allows us to do modeling and some of the stuff that I'm going to tell you. Yes?
So for a line and subplot.
So yeah, so the lateral line is formed by a series of these blobs. So each one of these green dots is one of these organs, and they're all connected by a line of cells, basically. And it's a very interesting process how this sort of develops. There's a patch of cells that walks under the skin of the fish and drops these organs in regular spacing. So there's a lot of work that other groups have done studying this process. So we basically study them once they're deposited, that's where we start the imaging. And yeah, so we can do segmentation, we can track the cells. Yeah.
So the line is formed by a group of different?
By a group of neuromasts. They develop individually, yes. I mean, it's a single organ in the sense that these are all connected by a chain of cells that maintain polarity and things like that. But you can consider each one of these as an independent organ. For the purposes of this talk, each one of these is an independent organ, and each one controls the number of cells in an independent way. And the development, so you can ablate one of these, completely kill it, and the other ones will do their own thing. And you can perturb cells in one of these, and the other cells don't realize. Yeah. So the other thing that makes it interesting for studying is that it's made by only about 60 to 80 cells during development, and there's only 3 cell types. So there's the mantle cells that form sort of like the periphery and give structure to the organ. The support cells, so these are stem cells that proliferate and make the other 2 types. And then the hair cells, which are the mechanoreceptors. So, and these cells are analogous to the ones that you have in your ear. So that, to me, sort of like makes these organs sort of like, if you want to understand biological pattern formation and organogenesis, this is sort of like the harmonic oscillator or the hydrogen atom of organs. Like, there's nothing that is, I would say there are probably not many things that are non-trivial that are simpler than this. So it's sort of like a level of complexity where, so there's another thing that's going to be relevant in a second, but it's sort of at a mesoscale where like we can do a continuum description of the organ. So the individual cells are important, but we can still sort of like put all the cells in a computer and simulate what they're doing. No? So the questions that we try to understand while looking at this organ is, so how do the cells know how to build this? So the organ starts by a collection of cells that are all the same. So you start with a patch of like these orange support cells, and these cells start to divide, and then they differentiate into the different types. No? So from the perspective of a cell, these cells need to solve 3 problems. So first, they need to make the right types. So you start from the orange cells, they need to be able to differentiate into the 2 types. They need to be able to count the right number of cells of each type. So if you have an organ that has too many cells of one type or the other, it doesn't work. It's the same as you're trying to build a hand and you have more bone than skin. That's not a good thing. And then they need to locate the cells in the right locations. No? But they do this in a decentralized way. So the cells don't have eyes. They don't know what's going on at the other end of the organ. The only thing that they can measure is what comes through their membrane. So the information that they can integrate is local information about what's going on. But based on that local information, they're able to integrate these signals and run some distributed algorithm that builds this in a very robust way. So like you look at hundreds of fish, you look at hundreds of neuromass, they all look more or less the same. They all have more or less the same number of cells and the same ratios of cells. And not only that, this organ is able to regenerate from almost any perturbation you do. So this is something that makes it also for biology and medicine applications is that your inner ears, in particular the hair cells of your ears, don't regenerate. So you're born with a certain number and they only die. So you die with less cells that you were born with. So for the young people, if you go to concerts, like wear protection, because when you kill the cells, that's it. And so that's why like 70% of people above 60 have some degree of hearing loss. In the case of the lateral line, you can kill any combination of cells and the organ will regenerate to something that looks statistically identical to the starting point. So the cells are not only able to calculate a path to the final state once, they're able to calculate a path from any perturbation you make in any sort of direction in phase space to the fixed point of the system over and over. And so how do they do this in a robust way? We don't know. And like in general, we don't know what the properties of a system that's able to do this are. So this sort of like brings me to, so how do we study this problem? So there's multiple ways someone can think about this. So one is sort of like the one that for me as an IFISCO comes more naturally, which is think about this as a dynamical system. So you have a bunch of cells that are sharing information. You can write the equations, the dynamical equations of how the information sort of like propagates, or like how different variables propagate. And you can think about what are the equations that describe a system like this. So how do you build like in general, dynamical system that is so robust under perturbations, where like you set like 50% of the variables to 0, and then the system still finds the same fixed point as it was before. Like, do these systems have generic properties, and do these systems also these properties transfer to other organs and other species? So, for example, in the zebrafish, they can not only regenerate their lateral lines, they can regenerate their brains, their hearts, the fins, So are the rules that the cells are using to robustly regenerate the same across different systems? One can also think about this as a sort of a computer science problem. So you can think about sort of what is the information that the system encodes. Where is the information encoded? So like if you ask a biologist, they will say, oh, well, the information is encoded in the genome. But if you grab one of these cells and you put it on a dish, it won't grow a neuromass. No, so they only grow a neuromass when they have the right inputs and when they have the right partner. So meaning that the information is distributed. In a sense, a lot of the information is distributed in the interactions between the cells, but we don't know exactly how that is done. And so what are then the algorithms that the cells use to solve these trajectories and to find the final state? So how do they measure the state and where it's encoded and how do they find it? And how do they integrate? So another thing that is sort of like interesting is how they integrate this local information, which is what they can measure, to measure global properties of the system. So how does a cell that can only see what's happening locally know what is the total number of cells in an organ? And then one can think also about this in terms of like statistical mechanics. So if you look at everyone in this room, if you look at all the neuromass, we're all human beings, but we're all different, but we're all sort of like a macro state that is being a human being, in the same way that the neuromass are all in a macro state. That means this is a neuromass, so it's very, very clear. You look at all of them, the cells in a way, they know they're part of something that is a neuromass because they know when they need to stop growing. But we don't know what is the possible distribution of microstates that give rise to something that one can call a neuromass. And we don't know what are the macroscopic variables that describe these macrostates. So is it the number of cells? Is it the density? Are there sort of like order parameters that describe sort of like the spatial location, for example, of cells that describe what is a neuromass? And something I find very interesting also is that how do the cells that only have access to the microscopic variables sense the macroscopic state? So again, sort of how does the cell, so this is sort of kind of like if you had a gas where the particles can adjust their velocity to match target temperature, how do they do this? No? So if you only have local information, do you bump into enough particles and you measure, or like how do you do that? And how much do you integrate? And what are the limits into how much you can sense these macroscopic variables? As I said before, so going back a little bit to the biology, what we know about this organ is that this self-organizing process is coordinated by information that the cells pass to one another. And this is done using what are called signaling pathways. So these are sort of like molecular tools that the cells use over and over when they're building an organism, when they're developing. So these have like weird names because they come from many of these come from research in flies, but they're called Wnt, YAP, Notch, and FGF. So these pathways tell the cells, the supporting cells, when they need to proliferate and make more. And then there's this Notch pathway that tells the support cells when they need to differentiate and make a hair cell. So I'm going to talk a little bit about this, but most of the talk is going to be about this process, like the differentiation process. And I just want to say sort of like we know a lot about the kind of like molecular details of these pathways. And like how does the information go from the membrane of the cell to the nucleus and sort of like carries information, but we don't know a lot about how this information propagates across networks of cells. And this is like, I mean, you have your router at home and you know all the details of your router. You're the person who designed it. And then I come and I ask you like, oh, explain how an email works. And you can explain it based on what the router does, because emails are an emergent property of a collection of routers. And I think this is something maybe I don't need to say here, no? But if you want to study a system like this, what you need is to develop tools to read what is the state of the system and then write some information into the system so that you can see how the information propagates. So this is what we've been doing for the past 3 years in my group. So now we have tools to image this process dynamically at very high resolution for a long time. And then we can use segmentation and tracking to follow all the cells and then start sort of like building correlations on like, oh, when we see a cell dividing, we see it in this context and it's doing this and it's doing that. And so what we're building now, we're almost there. Maybe next time I come, I can tell you about it, is also tools to measure what is the activity of these signals. So when a cell is receiving or sending signals, and how do the dynamics look like. And then we use, of course, theory, mathematical modeling, and some machine learning to try to make sense of this information. And we can also do perturbation. So like right now, we can just go, for example, and kill individual cells and see how the organ responds. But we're also building molecular tools to change the activity of these signals so that we can basically inject information into the cells and then go from building correlations to building causal relationships between the signals and the state of the cells. So using this segmentation and tracking, so we can follow these organs and we can see sort of like how the developmental process works. And so what you see here is one of these organs that is developing. In light blue, you see new pairs of hair cells that show up. And in green, you see new pairs of support cells, okay? So if you look at these for long enough, so the first thing you see is that there's a hair cell pair that showed up here, and then the next one is showing up on the opposite side. So this is called the dorsal and the ventral side of the organ. So the first one shows on the dorsal side, the next one on the ventral, and then there's gonna be another division happening here that happens again on the dorsal side. So there's this alternating pattern of differentiation events that happen in these restricted areas, where the support cells show up mainly in this sort of like C-shaped region of the organ, and sort of like if you aggregate over multiple of these movies, what you see is sort of like the support cells proliferate mainly in these poles, but with some proliferation events happening in this C-shaped region, and then the hair cells differentiate in these poles. And you can look also at like when the events happen, and you see sort of like clearly this distribution where like there's a differentiation event that happens in the dorsal position, then ventral, the dorsal, and then ventral again, while the proliferation events happen sort of like stochastically. So if you look at the interval distribution, you see that the proliferation events have an exponential distribution, so they're more or less sort of like uncorrelated with one another, and the differentiation events happen with a very clear normal distribution with a peak at 15 hours. So if you look at one of these neuromass, the most likely thing you're going to see is like if you see a hair cell a pair showing up in the dorsal pole, 15 hours later you're going to see one showing up in the ventral pole. So what we're trying to understand now is sort of like how do these spatiotemporal patterns arise from these signaling events. So the first thing we try doing is there's a problem with doing biology is that the number of potential relevant variables is almost infinite. So we have 20,000 genes, and any moment in time, any of these genes can be important. So the first thing we wanted to see is like, can we tell something by just looking at movies of cells without knowing anything about the genes that they express? Can we tell something about what they're going to do in the future? Okay. So the first thing we did is like we use our segmentation to do sort of like some feature extraction, and these features are so positional information. So where are the cells within a kind of like register neuromass? What is this sort of like the cell shape and the fluorescence of the markers that we're using for the cells? So what is the volume, the surface area, like how bright they are under the microscope, and then look at network properties. So who is a neighbor of whom, and how many, for example, for the hair cells, how many contacts they have with support cells or mantle cells or other hair cells, and sort of like total contact, betweenness, centrality of the network. And so we're trying to see if like these features are in any way predictive of the future state of the cell. So the first thing is looking at these proliferation events, so support cells that make more support cells. And what you see is sort of like, if you look at kind of right before the division event, kind of you get sort of like a sanity check, a trivial result, which is that the most important features for prediction are the surface area, the volume, and the nuclear intensity, because the cells double their size before dividing. So the cells like blow up, they double their size, and then they cleave in the middle, and they make two cells of sort of like the original volume. So you see this volume doubling right before the division. So like this is trivial, but sort of it tells us that our random forest classifier is not going crazy. It's doing something that makes sense. But then if you look at 8 hours before the proliferation event, which is like, if you look at the organ, you don't know that the cells are going to proliferate. You can't tell. The algorithm, the random forest classifier can tell based on the contact of the support cells with the mantle cells, it can tell which ones are going to differentiate. So that's sort of like the most important feature that it looks at. And then we can do the same for the support cells that will differentiate into hair cells. And again, like if you look at right before, surface area and volume are the most important predictors. And then if you look at 8 hours before, what is most important is the contact with the support cells. So this tells us that the relationships between the cells are important features to predict what they're going to do. And you can say like, oh, well, yeah, maybe, I mean, your features are predictive, but your prediction is only 10%, so who cares? So you can look at the prediction accuracy. And for both cases, for proliferation and differentiation, we can predict with about 80% accuracy if a cell is going to divide or not, just by looking at these features. So this already tells us that we don't need to measure all this complexity of 20,000 genes on the cells. Just by looking at the relationships between the cells, it's enough to be very accurate in what they're going to do. And this sort of like brings us back to what I was saying at the beginning, that most of the information here is encoded in the relationships between the cells and not specifically in the internal state of the cells, which makes sense. So like if you have an emergent system, a system with emergent properties, the emergent properties are a consequence of the interactions and not so much about the internal rules of a system. So now let's focus a little bit more on these differentiation events and looking at how we can also use a dynamical systems approach to try to understand how this works. So what we know from a lot of years doing mutations to the fish and chemical treatments is that if you inhibit the activity of this pathway called Notch, the organs develop an excess of hair cells, okay? And then if you overactivate this pathway, you end up with a lack of hair cells, okay? So basically, sort of like long story short here, no Notch, more hair cells, a lot of Notch, no hair cells, okay? And so this Notch pathway is very interesting because it acts through a positive feedback loop. So what it does is that when you have two cells that are in contact with one another, they try to inhibit each other. So this is a positive feedback, it's a winner-takes-all mechanism that leads to a spontaneous breaking of symmetry. So if you have two cells that are identical, just small fluctuations will get amplified, and then you will get to a cell that has a high level of NICD, which is sort of like a measure of the activity of this pathway, and a cell that has low NICD. So you start with two cells that are almost identical, and they polarize to two different states. And this is a trick that biology uses everywhere to break symmetries in tissue. So this is the way that, for example, your brain develops. So like you have neurons and glia in the brain, and they develop through this symmetric mechanism. The hair follicles in your skin develop through this mechanism. It's like it's everywhere in nature. So so if you look at this is sort of like for two cells, kind of like easy to understand. So you have positive feedback, one goes up, the other goes down. When you have a lattice of cells, what happens, like you can write the equations here. So there are many ways of modeling this pathway. We use this because it's just a simple realization with just one equation. But basically what the equation says is that each cell will integrate the signal that receives from its neighbors, and then we'll compare it with its own signal. And then this is sort of like filtered by a sigmoidal function that I'm going to show, like I should have it here, but I'm going to show it in the next slide. And then it decides on how much it regulates its own activity of the pathway. So if you do this in a in a lattice that is sort of like a regular hexagonal lattice, and this is sort of like the sigmoidal filtering of the signal that you receive from your neighbors. This system spontaneously breaks symmetry and creates a pattern of wavelength 1. No, so you get cells that are high, cells that are low, and this again, like this is how sort of like if you look at your hair follicles in your skin, they have a semi-hexagonal arrangement that is determined by a variation of this system. So the question now is, how does this work in the neuromass? So the neuromass is not this nice hexagonal arrangement of cells. The cells not only that, so the arrangement of cells changes with time, changes all the time, because the cells are moving around and exchanging neighbors. So when you think about how it will look, it looks like a network of cells. And so using our segmentation data, we can compute this network. So we can look at which cells are in contact with other cells, and this coupling between the nodes of the network is proportional to the amount of contact between the cells. So what we can do now is if we can sort of like simulate these same equations in this network topology. But if you remember the movie that I showed you, this network topology is changing because the cells are moving around, they're exchanging neighbors. So we can calculate the topology at every frame of one of experiments and use that as the input for the coupling matrix for the model, and then run the model and calculate what is the expected activity of this pathway across the organ. And you see some cells in gray here because we know from genetic data that these cells basically have a very high level of signaling, and they don't move depending on what happens around them. So they're kind of like setting the boundary conditions for the system. So when we run the simulations, for example, in this example, what you see is there are a lot of cells that are sort of like in this high notch state, but there are some cells that undergo these excursions and suddenly become low on notch and then become high on notch again. So depending on how the network changes, there's the network kind of like drives the change in dynamics of the cells, okay, and the cells sort of like follow the changing in the network. So what we can do with this is we know that the cells that undergo these low notch excursions are very likely to differentiate. So we know this from what I told you before. Like if you have low notch, then you differentiate into a hair cell. So what we can do then is to make it simpler, we can just label all of the cells in white, and then we'll label in yellow the cells that undergo these long excursions in notch signaling. And so what you're going to see here is that the cells show up in the poles where the differentiation events happen. So they don't show up throughout the neuromass. They only show up in the dorsoventral poles, and they show up right before a differentiation event happens. So that cell was yellow a few minutes ago, and then it went and differentiated. And when a cell differentiates, what happens is that these new cells inhibit the neighbors from undergoing another differentiation event. So now the cells that are undergoing these excursions show up in the ventral pole. And then you just follow them, and I think like this cell here, so it's going to get circled in a second, and that cell is going to undergo differentiation. So I think it's this cell here. So, and this is going to happen a third time later. So what we see here is that basically, so the system is growing homogeneously, but then every time you create one of these differentiation events, you locally inhibit your neighbors, and you prevent them from differentiating. So what the system is doing is using notch signaling to basically read the topology of the network, and the cells use this to decide when the local ratio of cells is off, then they will undergo, so they will coordinate with one another. So that's why you see multiple cells that are undergoing notch. So they sort of like, they try to inhibit one another. One of them will win, and it's the one that's going to differentiate, and it's going to inhibit the rest. So in this way, you prevent all the cells from suddenly differentiating and making, messing the ratio. But every time the ratio is off because you made too many support cells, you correct by having differentiation. So in a robust way, this sort of like winner takes all mechanism is able to kind of like read the topology of the network and make the cells react to it. So we can do some sanity check with this of what happens now if we change the global coupling of the system. So now we say, okay, the signaling, I'm inhibiting the signaling. So even if you're connected with your membranes to your neighbors, you can't pass signals, no? So the simulations say that, like, as you would expect, the system becomes less and less coupled, and then the number of Hertz cells increases, okay? Because every cell basically that doesn't have anyone to signal with, so they think they're on their own, and then they undergo differentiation. And we can do this experiment using an inhibitor of this notch pathway, and what we see is that qualitatively we can recapitulate the increase in the number of Hertz cells. So we can do this experiment, so we can look at these are the control neural mass. So you see, like, there's a bunch of Hertz cells surrounded by support cells, and then when we treat them with this inhibitor, they all become Hertz cells basically, so they get overrun. And what we see is, like, as we expect, they get the number of Hertz cells increases at the expense of the support cells. So it's the support cells that are being converted into Hertz cells because they can't see their neighbors anymore. And we can take movies of these, so we can look at the dynamics. And what we're trying to do now is to simulate the dynamics to see if there's any sort of like rhyme or reason to the temporal progression, also of how the cells start differentiating. And you see that you get a few Hertz cells at the beginning, and then, like, suddenly the whole organ is overrun by support cells, sort of like going crazy and differentiating into Hertz cells. And if you look at many of these, as you would expect, if you average over many of these events, what you see is that tight spatial coordination that you saw at the poles at the beginning of the talk is completely lost. So, like, the system becomes more and more sparse, and then it loses spatial coordination, and the cells, they all basically become independent. So what we're trying to do now is to test a little bit using molecular markers for this pathway. So this is delta, which is one of the molecules that instantiates the pathway. And you see, as you would expect, that in this case, there's this cell that is high in delta, and there's like a few cells that have a little bit of this molecule, which means that this cell is very likely to be the one that is going to differentiate if this. So you need to kill the embryos to do this imaging. So if these neural mass continue to evolve, this is very likely the cell that would have differentiated. And what we're doing is also using our segmentation algorithm to quantify these. And what we want to do now is to run the simulations on these snapshots and see if the simulations actually predict the right cells, no? Like over multiple of these neural mass. And just to finish, the other thing that we're trying to do now is to do perturbations to the system. So we can go and kill cells. So where there's this red circle, there was a red cell that we went and killed with a laser. And then we can see how the system responds. And what you would expect here is also that when you kill one of the Hertz cells, one of the surrounding support cells is going to sense that the ratio of Hertz cells to support cells is off, and it's going to differentiate. So what you see here is that there was a support cell, this one here, that was in contact with the dead Hertz cell, and now it went on and differentiated. So what we want to do now is to, again, run our simulations on these data and see if we can predict the right Hertz cell, or at least down to 2 or 3 cells, predict what's going on. Yeah, with that, I would like to thank my group, in particular Aquila and Tiger, who did most of the experiments, team at EMBL who did the simulations, and Suben also, who did the random forest classifier of the cell shapes and cell properties. And thank you for your attention.
Thank you very much for very, very nice talk. Is there any question? Well, I'll have one to start with. This the symmetry breaking, it was very. So when I saw that at the beginning that the first cells differentiate at the pole, and then at the other pole and go on, I thought that maybe this had to do with something like an instability at the system size. No? So,
yeah.
Then I thought I have to ask this. But then you continue, and then you show that this is actually happening because of there is an imbalance in the inhibition
There's, I mean, what we think is happening, but there might be, so you might be right. And so we're still trying to wrap our heads around exactly what's happening. So let me go back.
Yeah,
So here, for example, so what we think happens is that, so this system has already a broken symmetry when we start the imaging, which is that this pair probably is the last pair of cells that divided. And then these bias the divisions because now there's and this end of the neural mass, there's less Hertzl to support, the Hertzl to support ratio is lower. So the most likely place where the next division is going to happen is at the top. So the question is what breaks the initial symmetry? And we think that might be just random because if you look at neural mass, the sort of like you have both phases. So you have the phase where like is ventral first or dorsal first. We think that's what's going on, but I'm not 100% sure that there are no like organ scale. So one thing that we don't have to look at that, and we're building tools to look at it, is that there are two timescales in the system. So one timescale is given by how fast this coupling matrix changes, and that is given by the experiments. But then there's this timescale, which is the timescale of how fast cells reach a steady state when you put them together. No, so the signaling rate, and we don't know what this is. And depending on how you choose this parameter, you get different answers because you get in one extreme, the network evolves much faster than the signals. So the signals never reach a steady state. So basically like the network is doing what it wants and then the signals, they just can't catch up. In the other extreme, the signals equilibrate much faster than the network. So at every point in time, everything is sort of like adiabatically at steady state. And then the regime where we are, which is the one that we need to set the system to be able to predict which cells are going to differentiate, in which these two scales are similar. So there is a chance that signals do propagate from neighbor to neighbor. And we're still looking into it. Yeah, so there might be sort of like, I don't know how, because we're looking at this network that has a sort of its random properties. I don't have a very good way of like looking at these analytically, but I don't know. We can discuss some more. Yeah.
Thank you. Emilio first.
Yeah, the network is you take it from experiments, the network of contact, but the network is depends on how the cells move and how they create. So any attempt to what determines the network? What determines where the cells move? And where do they accumulate? Do you have any idea? Or
yeah. So we're working on these and sort of like, so the other, I guess, like, yeah, the next problem is, as you very well said, is like, where is this network coming from? And most of what generates the network is these proliferation events. So let me show it here. So it's these proliferation events. And so you see that the proliferation happens sort of like it's biased towards the head of the fish, and meaning that over long time scales, because if you look at the neural mass, it's fairly symmetric. Over long time scales, there has to be some cell flow from this end to this end, but it seems to be very slow, and we don't capture it. You know, even though we image for like 4 or 5 days, we don't capture very, very pronounced flows. So we don't fully understand sort of like how once you make, first of all, why do you make cells only here? Is there any rhyme or reason of where the cells show up? Because that locally changes the network. And then are there sort of like organ scale flows? And we know sort of like what the pathways are, and we're starting to look into it. But I think this is like, yeah, this is the next question we want to explore. Mm-hmm.
Yeah. So with a related question, I still don't understand in this network, in this network that you have sort of strong wings and weak wings. So you put some wings thicker than others. How that is measured, or is that? I mean, what do you measure? Or is that the result?
Yeah, no. So the strength of the links is how much membrane do two cells share. So I'm showing these. The cells are not touching here because just for illustration purposes. But in the microscopy images, so if you look at this image here, for example, the cells are all like touching each other. So what we can do is we expand these segments to fill the boundary, and then we calculate how much
is the overlap?
due to cells, and that is the weight of those contacts is how much membrane the two cells share.
But and then in the model,
Mm-hmm.
this is used, I guess, for the coupling between the different
Yes.
elements. But I guess that since this, when there is, for instance, a duplication, or this changes, so the
Yeah.
size of these rings
Mhm.
is a dynamical thing.
Yes.
So do you include that dynamics in the model, such that the model predicts how also these dynamic changes, or you put the measure dynamics on the model?
Put the measure dynamics of the network in the model. Yeah. So the model is so it has this coupling term here. You get it? So let's say this coupling term, this changes according to experiment. So
So you put by hand, but you
Yeah.
could, in principle, model also, I guess, this coupling strength, no? Because the overlap between cells is a consequence
Yeah.
say, of some geometrical consequences
Yeah.
of one cell moving or whatever, no?
So the model has these sort of like the coupling strength is a parameter that we can adjust. Yeah. So when we look at
But not only that, I'm saying that the size of the C sub j, the variation in time of the C sub j could be somehow also
well,
introduced.
I mean the variation we measure. So we measure the
Yeah, but-
CIJs.
But what I'm saying is that not, okay, you measure, but you
Yeah.
could introduce as a new variable and you put a model, you couldn't.
Yes, the
develop
problem is
a-
like I guess it's like what Emilio was saying. So we don't know how the network changes. Yeah.
Okay.
So yeah. So depending on what you assume, so we've done like models for like hard spheres, for example, for the cells, and like yes, maybe, but like how do you compare that to the real network? How do we know that? Because the network then is what actually determines where the cells are going to differentiate. So if your network model is wrong, then whatever you're gonna calculate from that is wrong. So that's why we're using the real network. Yeah. Mhm.
Not an attempt to say the dynamics for that.
Yeah. Yeah. We don't know what they are, and I think they might be very complicated because they involve cell mechanics, and they might involve also like global mechanics of the organ, and the cells do move according to their notch state also. So we know that, and so it's complicated. So yeah, so this is a way to like abstract ourselves from all those complications. It's like we just measure the network, and we look at the dynamics. Yeah. Mhm.
Just one final question then. When does the neuromaster stop growing? You didn't say.
So this is something we're still measuring. So these images that we are taking are between 2 days and 5-day-old fish, about 5-6-day-old fish. And this is where like most people have reported at 5 days, they say like, oh, the neuromass reaches a steady state. But what we found is that there's a very like slow residual growth until they're like 14 days. So like until they're 2 weeks old, they keep making cells very, very slowly. But this is a good approximation of the steady state. So this is sort of like at the end of the movie, they're almost final size.
But when they decide to stop, is something that is not in here, is something else that is going on, or is
So
in the dynamics
we don't
range?
know. So we don't know what it is. So we know that one of these pathways, Wnt, is involved in this because we know if we overactivate this pathway, the neuromass grows like 50% more in size. So the cells go crazy and start making more support cells. But there's also another pathway that's called YAP that is mechanosensitive that is also involved. And what we think might be happening is that there's a combination of some chemical sensing plus some mechanical sensing of like the skin is constraining, and so when the density gets to a certain level, so there's two things. So one is there's a measurement of the density, and then the other pathway says sort of like, okay, what is that threshold, no? And so if you lower the threshold, they grow larger, and if you remove some of the physical constraints, they will grow larger. Yeah. Mm-hmm.
One final question.
But so initially there's one cell, right? Or how does this?
Well, so the neuromass get deposited by there's a cluster of cells that sort of like walks along the fish, and then it loses pieces of itself. So when the neuromass starts, there are about 10 cells, in when they do this, but you can make tricks to the fish so that they build neuromass from single cells. Yeah.
I meant the entire embryo, right? Initially, it starts from one.
For the embryo, yes,
Yeah.
yeah.
So, but then when can you tell? So, in where head and tail are, it's sort of random, right? If you look at the first cell, you can't
Yes.
say- how many cells do you need to see to be able to say this will be the head and this will be the tail? Or no, this is sort of random, I assume, right?
So there's
The symmetry breaking.
yeah, so there's a very stereotypical process called gastrulation that happens when the embryo is about 1,000 cells. That is when the symmetry breaking happens, and it's a spontaneous symmetry breaking event. So like which side of is going to be end up going be the tail or the head is random. Yeah.
So okay, then my next question. So this is something I've wondered for a long time, and I don't understand. But so is there a chiral symmetry in this fish? So we have a chiral symmetry, right? You have your heart on
Yes.
the left, right? So why do we not have half of us the heart on the right and the other on the left, right? If everything is random, the symmetry breaking, you would expect.
Well, yeah, because it's not random. So like for fish, for humans, it's the same. So there's I mean, this is very, very coarse.
This has nothing to do with you.
No, but this is very coarse grained. So like, yeah, don't quote me on this. But during embryonic development, there's this in zebrafish thing called the Kupfer vesicle. So it's like a small sack of water where there's a lot of cells like facing inwards, and these cells have cilia that beat. And those cilia are made by tubulin, and tubulin is chiral, so they only beat in one direction. And when they beat in that direction, they create fluid flow that breaks the symmetry and always breaks the symmetry in the same way. So if you have mutations in the microtubules that mess up with the cilia, you can end up with situs inversus, or you can end up with situations where you get the heart on one side and the kidney on the wrong side respect to the heart. So like if you get situs inversus, that's like you can live a normal life and never know until you get an X-ray. If you get some like the organs distributed in the wrong, that can be fatal. Yeah. So. Yes.
Thank you very much.
Yeah.
Thanks a lot.
Gracias.
Es que, tú has dicho, puedo hacer un approach de statistical
Sí.
y después, pero no has hecho nada de eso.
Bueno, todavía no es algo que queremos hacer. Sí, es como